Hello there, and welcome back to Science for Sleep. Tonight we are going to explore something that is happening to you right now. Not in the obvious sense, not the breathing, the heartbeat, the slow release of the day's tension from muscles that have been working since morning. Something more hidden than any of that. Something that will continue through the night, through every stage of sleep, reshaping the neural architecture of your brain in ways you will not remember and cannot observe. Tonight, we are going to explore what happens to your memories while you sleep. Memory is the thread that connects you to yourself. Every relationship you have depends on it. Every skill you possess was built through it. Your sense of who you are, the continuous narrative of a life lived from the inside, exists only because of the extraordinary capacity of the brain to preserve experience across time. Without memory, there is no yesterday. No context for today, no sense of a self that persists through change. And yet memory, for all its importance, is extraordinarily fragile. The experience of a day, the conversations, the moments of learning, the emotions felt and the problems encountered, exists initially in a form so delicate that a single night of poor sleep can measurably degrade it. The difference between what you learned yesterday and what you will be able to use tomorrow is determined, in very large part, by what happens inside your brain tonight. Scientists have known for centuries that sleep and memory are connected. The folk wisdom that one should sleep on a problem before deciding, that things will look different in the morning, that a good night's sleep before an exam is worth more than a late night of cramming, has been present in human culture for as long as sleep and memory have existed together. What has changed in the last 3 decades is our understanding of why. The mechanisms, the stages, the precise choreography of cellular and molecular events that transform the fragile traces of a day's experience into the durable architecture of long-term memory. Tonight, where are you listening from? Are you already in bed? Is the room dark around you? Then your brain has already begun its preparations. The chemistry of wakefulness is shifting. The adenosine that has been accumulating in your brain all day, building the pressure of sleep, is beginning to find its receptors. The temperature at your body's core is beginning its nightly descent. The architecture of the night is assembling itself, stage by stage, in the neural systems that govern the transition from waking to sleep. And when you cross that threshold, the memory work will begin. So let your breathing slow. Let the weight of the day settle. And let's explore together what your brain is about to do. Let's begin. The brain faces a fundamental problem. Every day it is flooded with experience. Sensory information pours in from every direction. Conversations, encounters, observations, emotions, decisions, errors, corrections, moments of pleasure and frustration and surprise. The brain encodes all of it, to varying degrees, in patterns of neural activity, and it must somehow decide what to keep and what to discard, how to organize and integrate new information with existing knowledge, and how to preserve important experiences in a form that will be accessible and useful, not just tomorrow, but years, even decades from now. This problem is not trivial. The human brain contains approximately 86 billion neurons, connected by approximately 100 trillion synaptic connections. But the capacity of any individual neural circuit to hold information is finite. If every experience were preserved with equal fidelity and equal permanence, the system would rapidly saturate. New learning would interfere with old. The signal would be swamped by noise. The very richness of human experience would overwhelm the neural systems trying to record it. The solution the brain has evolved to this problem is a 2-stage memory system. The first stage is the hippocampus. This curved, seahorse-shaped structure, buried deep in the temporal lobe on both sides of the brain, functions as the brain's rapid acquisition system. When something happens to you, the hippocampus rapidly encodes the experience, binding together the disparate elements, the visual details, the sounds, the context, the emotions, the sequence of events, into a coherent, retrievable whole. It does this quickly and efficiently, designed for the demands of an animal moving through a constantly changing environment that must track locations, recognize individuals, remember dangers, and recall where food was found. But the hippocampus is not designed for permanent storage. It is, in the language of computing, more like RAM than a hard drive. It holds information in a high-fidelity but relatively fragile form, accessible for retrieval but vulnerable to interference and decay. The hippocampus is the staging area, the temporary holding space where new memories wait for the process that will transfer them to their permanent home. That home is the neocortex. The vast outer sheet of the brain, the rippled grey matter that covers its surface, is the brain's long-term storage system. It holds the accumulated knowledge of a lifetime, the semantic memories of facts and concepts, the procedural memories of skills and habits, the episodic memories of personally experienced events, organized in distributed networks that span multiple cortical regions and are gradually built up over years of experience. The transfer from hippocampus to neocortex, called systems consolidation, is the central process by which new memories become permanent memories. And it is a process that happens predominantly and most powerfully during sleep. The architecture of a normal night of sleep is not uniform. Sleep is not a single state. It is a sequence of stages cycling through the night in a precise pattern that reflects the different biological functions each stage serves. The early part of the night, the first 3 to 4 hours, is dominated by deep, slow-wave sleep, the stage characterized by the large, rolling delta waves that sweep through the cortex at 1 to 4 cycles per second. This is the stage in which the body's most urgent restorative work is performed. Growth hormone is released. Cellular repair begins. The glymphatic system dramatically increases its activity, flushing metabolic waste products from the brain tissue. And the first, most critical phase of memory consolidation begins. During deep, slow-wave sleep, the hippocampus replays. This replay was first directly observed in rats by the neuroscientists John O'Keefe and Lynn Nadell in the 1970s. And its role in memory consolidation was gradually established over the following decades. When a rat navigates a maze, specific neurons in its hippocampus, called place cells, fire in sequence as the animal moves through different locations. The pattern of firing encodes the spatial map of the environment. When the rat subsequently sleeps, these same neurons fire again, in the same sequence, but compressed in time. The navigation of a maze that took minutes is replayed in seconds. The hippocampus is running the experience again in fast forward in the quiet of sleep. The same phenomenon has since been observed in humans. Using electroencephalography and intracranial recordings, researchers have found that the hippocampus generates sharp wave ripples during slow-wave sleep, brief bursts of high-frequency neural activity that represent compressed replays of waking experience. Each ripple lasts only a fraction of a second, but in the course of a night of sleep, hundreds of ripples occur, each carrying fragments of the day's experiences from the hippocampus toward the cortex. The cortex is not simply a passive receiver during this process. It participates actively in a dialogue with the hippocampus, a dialogue orchestrated by the slow oscillations of deep sleep. The slow oscillation has two phases: an up state, in which neurons throughout the cortex fire in synchrony, and a down state, in which they fall into near silence. The hippocampal sharp wave ripples occur preferentially during the up states of the slow oscillation, when the cortex is maximally receptive. And embedded within each ripple, nested within the cortical up state, are sleep spindles, those brief bursts of activity at 12 to 15 Hz that mark the moment of transfer. This triple synchrony, the slow oscillation providing the rhythmic framework, the spindle opening the window of cortical receptivity, the hippocampal ripple delivering its payload of memory content, is the precise mechanism by which the brain transfers the experiences of the day into the structures that will hold them for a lifetime. Each cycle of the slow oscillation creates a brief moment of coordination between hippocampus and cortex. Each spindle within that moment carries a fragment of remembered experience. Across the course of a night, The slow oscillations occur thousands of times, the spindles occur hundreds of times, and the ripples occur hundreds of times more. The cumulative transfer is substantial. What was encoded in the hippocampus during the day is being distributed across the cortex during the night, linked to related knowledge already stored there, integrated into the fabric of long-term memory. The brain does not replay everything that happened during the day. It selects. This is one of the most important and most fascinating aspects of sleep-dependent memory consolidation. The hippocampal replay is not a faithful recording of everything that entered the hippocampus during the day. It is an edited version, shaped by signals from multiple systems that evaluate the importance, relevance, and emotional significance of different experiences. Emotion is one of the most powerful determinants of what gets consolidated during sleep. The amygdala, the brain's emotional processing center, communicates closely with the hippocampus during both waking encoding and sleep replay. Experiences that are emotionally significant, whether positive or negative, are tagged during encoding by elevated amygdala activity that increases the strength of the hippocampal memory trace. During sleep, these emotionally tagged memories are preferentially replayed and preferentially consolidated. They receive more spindling, more cortical integration, and ultimately more durable storage than emotionally neutral experiences. This is adaptive. The most emotionally significant experiences of a life are generally the most important to remember. The events that produced fear, joy, grief, or love are typically the events most relevant to survival and well-being. The brain's prioritization of emotional memory during sleep reflects the evolutionary logic of a system designed not for completeness, but for relevance. Novelty is another powerful determinant of consolidation priority. Experiences that are surprising, unexpected, or that represent genuine new learning are more strongly consolidated than experiences that confirm what is already known. The brain is not simply storing everything. It is investing its consolidation resources where the return on investment is highest, in the experiences that update the model of the world rather than simply repeat what the model already contains. Relevance to current goals also influences consolidation. Research has found that learning that is relevant to a person's current concerns and intentions is more strongly consolidated than learning that is disconnected from what they care about. Telling research participants before sleep that they will be tested on specific material without any additional study can increase the consolidation of that material overnight, simply because the information has been flagged as relevant. This means that what you think about before sleep can influence what your brain chooses to consolidate during it, not through any mystical mechanism, but through the very ordinary process of directing attention and flagging information as currently relevant. The long-standing advice to review material before bed, not to study it intensively, but simply to remind the brain of what matters, has a genuine neurological basis. The brain listens to signals about what is important, and sleep is when it acts on them. The question of what dreams are for has occupied human beings since the earliest records of human thought. Every culture that has left written records has grappled with the meaning and function of dreams. The interpretations have varied enormously, from divine communication to unconscious wish fulfillment to random neural noise. What modern neuroscience has added to this ancient conversation is not a final answer, but a more precise and empirically grounded set of questions. During REM sleep, the stage that dominates the later hours of the night and in which the most vivid and narrative dreams occur, the brain enters a state of high activity that in many ways resembles waking more closely than it resembles deep sleep. The prefrontal cortex, the seat of rational evaluation and executive control, remains relatively suppressed, which accounts for the uncritical acceptance of impossible scenarios that characterizes most dreams. But the hippocampus and the limbic system, including the amygdala, are highly active. The default mode network, the brain's system for self-referential thought and imagination, is engaged. and the visual and motor cortices fire in patterns that mirror waking perception and movement, even in the absence of actual sensory input or physical action. This neural profile supports a specific role for REM sleep in memory processing, distinct from the role of slow-wave sleep. Where slow-wave sleep appears to be primarily concerned with the transfer of factual and episodic memories from hippocampus to cortex, REM sleep appears to be more concerned with the integration of new memories into the broader fabric of existing knowledge, and with the processing of the emotional content of memories. Matthew Walker's research at UC Berkeley has produced compelling evidence for what he calls the overnight therapy function of REM sleep. During REM, the brain reactivates emotionally significant memories in a neurochemical environment that is dramatically different from waking. Norepinephrine, the brain's primary stress neurotransmitter, is almost entirely absent during REM sleep. This is the only period in the 24-hour cycle when norepinephrine drops to essentially zero. The result is that emotionally charged memories are reactivated and processed in a state of reduced physiological stress, allowing the emotional charge to be gradually separated from the factual content of the memory. This decoupling of emotion from memory content is, as Walker argues, what allows the sleeping mind to revisit painful or distressing experiences without being re-traumatized by them. The memory of a difficult event, replayed during REM sleep over multiple nights, loses progressively more of its raw emotional charge while retaining its factual and contextual content. We remember what happened without being overwhelmed each time by the feeling of it happening again. The dreaming that accompanies REM sleep may be a byproduct of this process, the subjective experience of the hippocampus and cortex exchanging and recombining memory fragments in a state of reduced executive oversight. The narrative quality of dreams, their tendency to combine elements from recent and distant memory in unexpected ways, may reflect the brain's active search for connections between new experiences and the existing knowledge structures that give them meaning. Some researchers have proposed that the bizarre juxtapositions characteristic of dream content, the sudden transitions, the impossible combinations, the appearances of people and places from different periods of life, are not random but serve a specific computational function. By combining disparate memory elements in novel ways, the dreaming brain may be testing potential connections, identifying relationships between experiences that the waking mind, constrained by logical consistency and sequential attention, would never have the opportunity to explore. The researcher Robert Stickgold at Harvard Medical School has found evidence for exactly this kind of creative memory integration during sleep. Subjects trained on a task and then allowed to sleep showed subsequent performance improvements that went beyond what they had actually practiced, accessing deeper structural features of the task that had not been explicitly taught. The sleeping brain had found patterns in the training data that the waking brain had missed. There is a paradox at the heart of sleep and memory. Sleep is when the most intensive memory processing of the day occurs, yet we remember almost nothing of it. The amnesia of sleep is nearly complete. We wake with no memory of the slow oscillations that swept through our cortex, no record of the sharp wave ripples that carried the day's experiences from hippocampus to neocortex, no awareness of the spindles that opened the windows of cortical receptivity, no recollection of the thousands of synaptic changes that consolidated the learning we will use tomorrow. The most important memory work of the day leaves no memory of itself. This is not an accident. It reflects the functional requirements of a system designed for transfer and integration rather than for recording its own operations. The hippocampus, during slow-wave sleep, is replaying experiences rather than encoding new ones. The prefrontal cortex, which normally supports the formation of autobiographical memories by providing the contextual framework within which experiences are located in time and place, is suppressed. The conditions for the formation of explicit, recallable memories of the sleep process itself are precisely the conditions that are absent during sleep. The dreams we do remember are the exceptions, the fragments that survive the transition to waking, usually because we wake directly from REM sleep and carry some residual activation of the dream content into the waking state. but they represent a tiny fraction of the night's total dream production. For every dream fragment recalled upon waking, dozens or hundreds of others have dissolved without trace. This amnesia protects the coherence of waking memory. If the brain recorded the sleep process in the same way it records waking experience, the memories of sleep, the fragmented and often impossible scenarios of dreams, the absence of temporal and spatial context, would interfere with the structure of waking autobiographical memory. The night's processing would contaminate the record of the day. The consequences of disrupted sleep-dependent memory consolidation are not subtle. They are measurable, consistent, and cumulative. A single night of total sleep deprivation before learning produces approximately a 40% reduction in the brain's ability to encode new memories, as measured by subsequent memory tests. The regions that show the most dramatic impairment are precisely those most involved in the initial encoding of new experiences, the hippocampus and the prefrontal cortex. A night without sleep does not simply make you tired. It degrades the neural systems that memory formation depends on. But total sleep deprivation is rare in ordinary life. More common, and in some ways more insidious, is the chronic, mild sleep restriction that affects enormous numbers of people in modern societies. Sleeping 6 hours a night instead of 8, night after night for weeks, produces deficits in memory and cognitive performance that accumulate gradually and consistently. What makes this particularly concerning is that people who are chronically sleep restricted tend to lose awareness of their own impairment. They adapt subjectively to the feeling of mild sleepiness and begin to rate their performance as normal or near normal, even as objective measures of memory, reaction time, and cognitive accuracy continue to decline. The specific stages of sleep matter as much as total sleep duration. Disruptions that selectively target slow-wave sleep, the stage most critical for the consolidation of factual and episodic memories, produce selective deficits in those memory types while leaving other cognitive functions relatively intact. Disruptions that selectively target REM sleep produce deficits in the integration of new learning into existing knowledge structures and in the processing of emotional memories. Alcohol, which is commonly consumed as a sleep aid, is one of the most damaging disruptions to sleep-dependent memory consolidation. The sedating effects of alcohol ease the transition to sleep and increase the duration of slow-wave sleep in the first half of the night. But as alcohol is metabolized, it produces a rebound effect that suppresses REM sleep in the second half of the night, precisely when REM periods are longest and most important for memory integration and emotional processing. The net effect on memory consolidation is negative. Learning that occurs before an evening of drinking is less well consolidated by the following morning than learning under equivalent conditions without alcohol. Age-related changes in sleep architecture produce progressive changes in memory consolidation. The amount of slow-wave sleep declines substantially across the lifespan, with the greatest decreases occurring between young adulthood and middle age. Sleep spindle density, a direct predictor of overnight memory consolidation, also declines with age. Research has found that the age-related deficit in sleep-dependent memory consolidation can account for a significant portion of the age-related deficit in declarative memory that is one of the most widely noted features of cognitive aging. The memory is not simply failing to be stored. The machinery of overnight consolidation is becoming less efficient. There is a tradition in many cultures of sleeping on important decisions, of seeking in the morning the clarity that the evening could not provide. This tradition is not simply waiting for the emotional heat of a decision to cool. It is, at least in part, a form of intuitive wisdom about the brain's night work. The research literature is full of demonstrations that sleep does not merely preserve what was learned before it, it transforms it. The Hidden Shortcut study, conducted by Ulrich Wagner and colleagues in Germany, trained participants on a sequence of mathematical problems that could be solved by a tedious method or by an elegant shortcut that the participants were not explicitly taught. After a period of sleep, participants were almost 3 times more likely to have discovered the shortcut than those who spent an equivalent period awake. The sleeping brain had found the pattern that the waking brain had missed. Research on language learning has found that sleep following vocabulary learning produces not just better retention, but better generalization. The ability to recognize words in new contexts that differ from the training examples. The sleeping brain had extracted the abstract structure of the vocabulary, not just stored the specific examples. Studies of creative problem solving have consistently found that people are better at problems requiring insight, the sudden restructuring of a problem's representation that produces an unexpected solution after sleeping than before. And the improvements are not simply due to rest. They are specifically produced by the combination of REM sleep with the preceding period of waking with the problem unsolved. The sleeping brain continues to work on problems that the waking brain has set aside. Thomas Edison was famous for his habit of sitting in a chair with steel balls in his hands and a metal plate on the floor. As he drifted towards sleep and his hands relaxed, the balls would fall onto the plate with a crash, waking him. He would then immediately record whatever had been in his mind at the moment before waking. The same technique was used by the surrealist Salvador Dali. Both men had discovered, through experiment and intuition, that the threshold between waking and sleep was a particularly generative mental state, one in which the constraints of logical, sequential waking thought were loosening, and the free associative, pattern-seeking processes of the sleeping brain were beginning to operate. What they were harvesting was the early produce of the night's work. The creative, integrative, pattern-finding activity that the sleeping brain performs across the entire night was already beginning in the first moments of the transition. They had found a way to sample it before it dissolved into the amnesia of sleep. Those were the sleep science facts about how your brain processes memories while you drift into deep sleep. And now the processing can begin. Right now, as your breathing slows and your eyes grow heavy and the weight of the day finally settles into something that feels like permission to rest, your hippocampus is beginning its work. The sharp wave ripples are preparing. The slow oscillations, those long, sweeping tides that will roll through your cortex through the hours ahead, are building. The day is not over yet, not for your brain. The conversations, the moments of learning, the things that moved you or surprised you or required your attention are waiting in the hippocampus, tagged for transfer, queued for replay. The brain has been patient all day, accumulating what needs to be processed. And now, in the quiet of this room, in the slow release of everything that wakefulness requires, it has the conditions it needs. You do not have to do anything to help it. You never could. The consolidation proceeds with or without your awareness, following rules laid down over hundreds of millions of years of evolution, refined to a precision that no conscious intention could improve upon. What you can do, and what you are already doing, is give it time. The night is long enough. The stages will cycle through in their ancient sequence. The slow waves will build and crest and recede. The spindles will open their windows. The ripples will carry their cargo from the temporary to the permanent, from the fragile to the durable, from the experience of a single day into the architecture of a life. By morning, you will not remember any of it, but the person who wakes tomorrow will know things the person who fell asleep tonight did not. Will have connections where there were only fragments. Will find that problems that felt stuck have quietly loosened. Will discover that the learning of yesterday has become, overnight, something more than it was. This is the gift the night gives. Not rest, though it gives that too. Not escape, though it offers that as well. But a chance for the day's raw material to be made into something permanent. For experience to become memory, and memory to become the self. Let the slow waves come. Let the work begin. Science for Sleep will be here when you return.